Well, hello everyone, and welcome to this new series on genetics. So this semester I'm going to be teaching genetics, and I decided that I'm going to produce side content to go with my course to put on YouTube for everyone can see it. So I typically just record lecture videos using the lecture PowerPoints I have for my classes, but they contain a lot of copyright images, so I can't use those. So I plan to do these little side recordings based on this little outline I've put together here, and just, have some open access genetics, and you can follow along with the course. It's up to you how much you want to watch, but it should be a lot of fun, and I really want to produce more content like this for my channel. I know I started that anatomy and physiology, those videos like this with all, copyright free images and things like that. That's the hardest part about doing this online stuff open to everyone, is the copyright. But we'll see how this goes. So this one is going right into chapter 1 here, which is an introduction to genetics. So this is all just a big general overview of genetics and things we're going to be talking about, I'd say this semester, but in this series. So right now I'm teaching online mostly because of COVID. So I figured, why not share this with as many people as I can. Again, this isn't the direct lectures I do for my college. These are little side lectures I'm doing for my YouTube fans. All right, so let's get into this. So here, first, what I'm going to do is I go through this little outline here, and we kind of fill it in together and talk about the major topics. So for genetics, we of course have to define genetics. And genetics is the study of heredity, genes, and gene expression. So it's the study of heredity. Sorry, my handwriting is bad. And gene expression. So, genetics is massive. Heredity, passing certain traits, certain, diseases, studying that and passing them to offspring, chances of an offspring getting something, studying particular genes. So genes are little segments in our DNA that produce something like a protein. And then gene expression, what controls that regulation of expression. So importance of genetics. So genetics is obviously important to us. So that's biology, of course, because we are made from our genes. So that's, genetics underpins all of biology. We wouldn't have biology if it wasn't for genetics. And also the first one up here is individual or medical. So studying certain disease states and so forth. So individual, medical. So we have 20,000 genes in the human genome. So studying all those different genes in the genome, what happens when one thing goes wrong, and so forth. So this is looking at the physical traits, physiology of diseases, immune function, and even psychology as well. So individual, medical, and also one that we know, we've heard of GMOs. So industry and agriculture are another big importance. So industry / agriculture. I should just write ag. So, making better rice plants, things like that. Now, everyone, is slightly afraid of GMOs, but if done right, they're pretty advantageous, especially to third world countries. Industry, mass producing something like insulin, penicillin, things like that to make things cheaper, more easier for people to access and whatnot. So obviously genetics quite important in these disciplines. So how do we study genetics? We don't do all these genetic studies in humans, of course, because of ethics. You can't really control reproduction in humans ethically. And they have a long generation time, 9-month gestation period, and only one offspring. So for genetics is based on statistics. So this, of course, we're going to be getting into a lot of math and statistics, which a lot of people don't like, but I find exciting. So we use model organisms, things like C. elegans, which is a little nematode roundworm. We use mice, of course, E. coli, Saccharomyces cerevisiae, which is yeast, Drosophila melanogaster, which are fruit flies. All these are ideal model organisms. So what do we look for in these model organisms? So there's 6 main characteristics I like to mention. 1, short generation time. So you don't want to spend a long time figuring out whether your data is accurate or not. You want repeatable results. And to help with that, you want a high end value, so you want many progeny or offspring. You also want to be able to control genetic crosses. What does that mean? That means, okay, let's say we're, mixing fruit flies. This one has black eyes. This one has white eyes. What happens if we cross those two specifically? This one has regular size wings. This one has vestigial or smaller wings. What happens if we cross them? So you can control crosses in a maintained, simple way. Has to be cheap. So they have to be reared cheaply in a laboratory. Science is an expensive endeavor. So the cheaper you can make things, the better. Also, many genetic variants. So many variants available to assist in studying. So look at mice. There are tons of different forms of transgenic mice. So for my PhD research, I studied Alzheimer's disease. We used a transgenic form of a mouse that developed plaques in the brain. C. elegans has tons of different forms with certain proteins, enzymes turned on and turned off to make studying them a lot easier. And then you can study the different differences. We'll look at pea plants for Gregor Mendel's work, round versus wrinkled peas and so forth. Lots of different variants just in those pea plants. And then also well characterized. This is a pretty important one. So you don't want to be studying something that you don't know what makes it up. So like fruit flies, chromosomes are pretty well characterized in fruit flies. We know where all the genes are and so forth. All right, so that's just the importance of the model organisms. Now we can look at some of the major subdisciplines. Like I said, this is an overview of genetics in this course. So we're going to be going through each one. We'll be starting out the semester by talking about transmission or classical genetics. This is the Mendel stuff. We'll be doing Punnett squares. This is also where we get into statistics. So this is heredity and how traits are passed. So this one is looking at that transmission from one to another, looking at cell division, the chromosomal basis of inheritance. Remember, we always write, big A, little a. So for a dominant trait and a recessive trait, and you can have homozygous. So this is called heterozygous. This is called homozygous, where each chromosome carries the same trait, and then you can have recessive. So each one is, a lowercase a. That's typically how we represent that. We'll go over this in more detail coming up in a future chapter. But these are, remember, you get one of these from each parent. So one parent may give you the recessive form and the other parent may give you the dominant form. We can study that, do statistics on it, and so forth. That's, looking at the chromosomal basis of inheritance. You can also do gene mapping. So if we have a chromosome here, a rough idea of a chromosome, gene mapping is finding where that particular gene is on that chromosome and then linking them, and also finding out what other genes are located nearby or on another chromosome. So we'll be talking about that a little bit this semester too. Here the focus is individual organism and reproduction for transmission classical genetics. Next subtype is molecular genetics. This is my favorite area just because my background is more in molecular biology. So this is the chemistry of genes. So looking closer at cell division and how gene expression is controlled and regulated. So how come your beta islet cells in your pancreas produce insulin, but your muscle cells do not, even though they carry the gene for insulin in their genome? So we'll be looking at gene expression, regulation, and control. So the focus of molecular genetics is gene structure, function, and organization. And then last one here is population genetics. This is looking at more of evolution and conservation for the focus. So this is looking at genetic composition of a population, not of an individual. Genetic composition of a population. So looking at the total number of round versus wrinkled peas in a population. So talking about pea plants here. So for pea plants, you can have a round pea or you can have a wrinkled pea. And so if you're looking and the number of wrinkled peas are starting to increase in a population, you'd be studying population genetics, and you can look into maybe what could be causing that. I know that's a silly example, but something like that. You're looking at how things change over time within a population. So now the last little bit here is going over some key concepts in genetics. So I don't know where all your backgrounds are coming from while watching this, but this is just, we'll be saying these terms a lot throughout the semester and comparing them. So I just wanted to do a quick little summary of the major concepts here. The first, looking at organisms and viruses that contain genetic material. So going down all the way back here to the common ancestor of all life, we then formed three branches or three main domains. First one here were the bacteria, and then we had the archaea, and then we had the eukarya. So we are branching, from the eukarya, if I can spell it correctly, the eukarya, and then the archaea branch right before us. So the bacteria and archaea are known as prokaryotes, and the eukarya are known as the eukaryotes. So there are some major differences and similarities between prokaryotes and eukaryotes. So eukaryotes are you. That's one way to remember that. And so here, prokaryotes, again, the domains are bacteria and archaea. However, even though archaea is with the bacteria, some archaea are more similar to eukarya. So that's why archaea get their own domain, even though they are prokaryotes. So what are the major differences? One is, well, the biggest one is membrane-bound organelles. So here, membrane-bound organelles are absent, but in eukaryotes, they are present. So what do I mean by membrane-bound organelles? A nucleus. So if I were to draw a prokaryote here, all the DNA is circular and found just on the inside, and any other enzymes and things like that, like ribosomes, are found throughout this. Whereas a eukaryote, usually defined in a central region called the nucleus. So this is an internal membrane. Then you can have mitochondria, which are membrane-bound organelles. You can have the rough ER, you can have Golgi apparatus, and so forth. You have all these other membrane-bound organelles within a eukaryote. Also, here, just as an aside, we can jump to the last one down here, is size difference. Right here we have a mitochondria, my rendition of a mitochondria. A mitochondria fits into a eukaryote. A mitochondria is about the same size as a prokaryote. So relative size here, prokaryotes are smaller, much smaller, eukaryotes are larger. And then genome here, genome for the prokaryotes, it's a circular chromosome. So one circular chromosome. And then here it's 1 plus linear chromosome. So humans, 23 pairs of chromosomes. So circular versus linear as well. Histones are these little proteins made up of different subunits that DNA can wrap around, and it helps it package down into discrete units. So prokaryotes, bacteria, no. Archaea, though, some have histones. Eukaryotes, all are present. All eukaryotes have the histones. So that's prokaryotes for eukaryotes. Now viruses. So are viruses considered dead or alive? Trick question. You can't define a virus as dead because that would suggest that it was once living. So viruses are called non-living. So all a virus is a protein capsid plus DNA or RNA. So all it is a protein plus genetic material. There is no cell membrane around a virus. There are different structures of virus, and if I ever make a course in microbiology, we'll go over a lot more detail about viral structure and the different classes of viruses. But a virus is not considered alive. It's non-living. So why? So it's not alive because of a couple of different reasons. One, it has no metabolism. There's no enzymes doing anything in it. It can't produce its own energy. So because it has no metabolism, it can't do things. It can't replicate itself. So it's not made of cells. There's no phospholipid membrane. Also, no homeostasis. So when I walk outside into hot or cold weather, my body temperature can regulate itself so that if I walk out into freezing temps, my body doesn't become freezing temps. You start shivering to keep it warm. And the last part here, so that's homeostasis. A virus will become the surroundings. And then here, cannot reproduce. That's one of the big ones. It requires a host for reproduction. So the coronavirus goes into your cells, uses your cells' machinery to reproduce itself. All right, so those are the major classes there. And we're going to talk about some of those throughout the semester that contain genetic material. Now let's talk about some molecules that make up the genetic material. So we have the basics here. We have DNA, and we have our RNA, deoxyribonucleic acid and ribonucleic acid. The main difference here is the sugar that makes them up. This is a ribose. This is a deoxyribose. We'll talk more about the differences in the structure later. But here, genetic info in the nucleotide base sequence. So here, DNA has certain base pairs in it. So here, let me move that there. There we go. DNA is made up of adenine, guanine, cytosine, or thymine, whereas RNA, so these are the nitrogenous bases that make it up. This might be a review. You might be hearing these for the first time, so we'll go over it in more detail in a later lecture. RNA then is made up of adenine, guanine, cytosine, and uracil. So there's no thymine in RNA, and that's one major difference. DNA is also double-stranded. So double-stranded. RNA is single-stranded. So here, this was a strand of DNA, this would be a double strand, whereas RNA would be a single strand. That makes DNA more stable, and RNA is less stable. However, that allows RNA to have more diverse functions. So RNA can actually form, hairpins type structures. So it can have bonds going like this, it can form this little hairpin loop here, and have these varying structures throughout. So RNA can actually act as an enzyme in some cases when it's called a ribozyme. A ribosome is an example. So a ribosome is what takes your messenger RNA and turns it into a protein. Ribosomes are made of RNA and a little bit of protein. Okay, so more on the nucleic acid here. A chromosome, that's what the package is, the discrete units seen in your cells. That's DNA plus protein. So both of them make up a chromosome. The DNA wraps around those histone proteins. Then a little bit more about the definitions here. A gene, that's a basic unit of heredity. So genes, it's a section of DNA that codes for a product. So this could be, made up of code for a protein or code for RNA. When we talk about genes, we talk about a locus. A locus is the location of that particular gene. So you get stadium assigned seating if you go to a sporting event. That's how we define where that gene is as well. So it's the location of a gene on a chromosome. If I draw my little horrible chromosome here and I say that's where that gene is, we say it's on chromosome 5, we say it's on the P or the Q arm, and the location is away from the centromere. That's how we would define that. We'll talk more about the descriptors there later. A genome then, this is all the genes in an organism. So all genes in an organism. I'll just write all genes here to keep it simple. So the Human Genome Project was studying all the genes in the human organism that's been happening over the last decade. DNA replication is just simple terminology here. DNA replication is when you go from one DNA to two DNA. This is a vital process in evolution and the production of us. So you start out as one fertilized cell, and then you have to replicate your DNA into new cells going through mitosis over and over again to produce what you are today. You want all that DNA to be genetically identical. And then we have the central dogma of biology, which is how we make protein. So we go from DNA to messenger RNA. This process is called transcription. And then we go to protein, which is called translation. This is known as the central dogma of molecular biology. I remember it because nucleic acid and nucleic acid, same language, so it's a transcription, whereas here you're going from a nucleic acid to an amino acid sequence. So you're translating it from the language of nucleic acids to the language of nucleic acids. Okay, so now a little bit about genetics and some terminology used. One important terminology here are alleles. So these are different versions of genes. So here I'll just write different versions. So when I was talking about round versus wrinkled peas, so determining whether a pea is round or wrinkled is found in the same location. It's the same gene. But one could cue for round, one could cue for wrinkled. So in this case, round is dominant. So round could be big A, or big A, little a. That dominant form makes it round, whereas wrinkled is the recessive version and would be two lowercase a's. So these are different versions of the same gene. So this can look at then phenotype then. So when I'm writing these letters, I'm writing the genotype. So this is the genetic makeup of that particular allele. It's the allele combination, and that's what defines there. So a phenotype though is what you see. So this is the physical appearance of the gene. So here, these both are round if we're using the pea plant, whereas this one is wrinkled. Now, if I hand you a round pea and I say, what's the genotype of this? Can you 100% say which one it is? No, you can't. So phenotype, pheno does not always equal geno, unless it's recessive. If I hand you a wrinkled pea, you can be pretty sure it's a little a, or whatever letters you're picking. Okay, now also want to look at, so how do we go past things to offspring? We got to go through cell division. Your cells divide and produce new cells. Prokaryotic cell division, this all happens via a process called binary fission. So that's where you have your cell. So we have an E. coli here. It replicates its genome, the 2 circular chromosomes. It then splits in the middle and then produces 2 daughter cells via asexual reproduction. So binary fission. That's how most bacteria replicate. Eukaryotic, though, a little bit more complex. So we have first mitosis. So we have one parent produces 2 daughter cells, 100% genetic identity. So one parent DNA becomes 2 DNA, goes into 2 daughter cells, identical daughter cells. So that's mitosis. Now the other division we have is meiosis. Meiosis is where you have one parent. So we have meiosis 1 and meiosis 2. So it goes through 2 rounds of division and ends up producing 4 daughter cells that are 50% identical. So meiosis produces non-identical daughter cells. Mitosis produces identical daughter cells. Meiosis is really important in production of germline cells, so sperm and egg. So that's why offspring are slightly different variations of the parents. And it's because of that 50% genetic identity to the parent cell. All right, last little concepts here I want to go over. So how populations evolve by genetic change, mutations. Mutations happen in a population. So a mutation could happen in an individual, but if it doesn't present itself in a population, it doesn't matter. So the mutation has to be defined as a permanent and heritable change. So I could go out in the sun and my skin gets, DNA gets damaged, but if I can't pass that damage to an offspring, it doesn't matter. It's not a permanent heritable change. It's not something I can pass along. That's a mutation, and if that mutation is advantageous, it might end up presenting itself in a population, leading to evolution. So evolution is a change in frequency. So a change in frequency in a population of the alleles. So let's say we had a, let's stick with the round versus wrinkled peas. Let's say we started here, there's a little pie chart, and let's say here 25% were wrinkled and 75% were round. Now, let's say we go through a couple generations and something happened in the environment. So now the wrinkled pea pods, for some reason, are surviving a lot better than the round ones. So there's a drift, a natural selection event, so a change in frequency within the population. Remember, individuals do not evolve, populations evolve. So now we have 50% wrinkled and 50% round. So if you're looking at the actual alleles in that population, you can see a big shift to this recessive one. There's also one you can't forget about over there in the heterozygous form, whereas over here, they were much less. So we'll talk about that a little bit later as well. We'll be doing statistics and problem sets with that. But all right, that's all I have for this first chapter here. So this was a big, rough, and a little sporadic, jumps around a lot of different concepts, overview of genetics. But we're going to go into lots and lots of detail of all these topics throughout this YouTube series. Let's hope I don't get too crazy busy and I can keep producing these videos. I know this one's a little longer, about 24-25 minutes. Maybe the future ones I'll break up a little better so that you don't feel like you're sitting there as long watching. I don't know, let me know in the comments if you like the longer versions or want me to, separate them, and maybe I could have stopped, right here today and then talk about the DNA part in a separate video. So shorter, maybe 10-minute videos for these topics. But all right, that's all I have for today. Let me know if you have any questions in the comments. I look forward to producing this series for you, and I hope you enjoy it. All right, everyone have a great day. Bye-bye.